All right, folks, I guess we'll go ahead and get started. Welcome everyone to another session of our ADA Pathways seminar series. Really excited about the two talks that we're going to hear about today. Before we launch, I want to make a quick announcement. Chandra Gruber, the ADA's Director of Research Programs, has sent an announcement to all of you, but I just wanted to come behind that and emphasize an opportunity, which is a small group of MAG members and myself and Chandra picked out 4 speakers for an ADA Pathways Symposium, and that will be coming up on Friday, June 25th from 10 to 11. The details and registration information is in a recent email from Chandra. The speakers will be Judith Agudo, Paul Cohen, Marie-France Hivert, and Philip White. And in case you're wondering how they were chosen, we really tried to spread it out in terms of duration of award. So they're everything from veterans to newer awardees. We wanted to try to have representation of different flavors of diabetes research. So we've got pre-diabetes, type 1, type 2, and gestational diabetes represented in those talks. And wanting to be politically correct, we wanted to have a good mix of male and female. So we have 2 male and 2 female speakers. And of course, we didn't pick people who spoke at the event that we had last year that Magda and I were involved with. And so rest assured, if we're successful in this program at the ADA, where Chandra is going to be trying to invite people that might be supportive of investment in the program in the future, we will be calling on others of you to do this seminar, this program at next year's ADA, and I hope that that will be the case. And thanks to all of those that did accept our invitation and will speak, and I hope everybody can join us on June 25th, 10 to 12 Eastern Time. Okay, so our first speaker today is a friend, Praveen Sethupathy. I met Praveen when he was a faculty member at UNC Chapel Hill, just up the road. His award is an Accelerator Award, and the title is The Chromatin Activity Signature of NASH Resistance in Humans Reveals Key Candidate Drivers of Disease. Praveen.
Thank you, Chris, and thanks everybody who was able to jump on today. I've been a huge fan of Sarah and her work for some time now. So my goal here is to get through this and be able to really listen to her exciting work. But no, in seriousness, this is a very nascent project, but it's one that I'm excited about, and I'd love any feedback that you all have. Is very much ongoing right now. I'm currently in the no-cost extension period of my ADA Pathway Award. So I view this talk as kind of my pathway swan song, if you will. So I really felt that it was apropos to start with a summary of a couple of different projects that simply wouldn't have been possible without support from ADA Pathway. And I've said that many times before without giving concrete evidence of some of this work and publications. So I wanted to take the opportunity to do that now. And then I'll get into an intro to the main topic, which is NASH, as you heard from Chris. And then defining the chromatin activity signature in NASH-resistant humans, and some ongoing work and future directions. One of the first things that I had the opportunity to do with the ADA Pathway funds was to establish the tools and the techniques in my lab in order to be able to take advantage of systems genetics resources like the diversity outbred mice for the purposes of large-scale genetic mapping. Our primary interest was to try to understand genetic contributions to variability in phenotypes in the gut that we really cared about a lot that were relevant to diabetes. A prominent example would be the relative abundance and function of gut endocrine cells, which of course, as I think all of you know, secrete a cocktail of hormones that are critical for energy homeostasis. That study is still ongoing, and we hope to, in the forthcoming months, maybe a year, have something out on that. But what we realized is that with the sort of laborious work of setting up these resources, we could sort of feed two birds with one hand, if you will, and establish collaborations where we could look at all kinds of other phenotypes. And over a Thanksgiving meal with my sister, Priya, who is a PI at Rockefeller, we sort of birthed this idea to use the same resource in a lot of the informatic workflow that we had set up to be able to help her identify what turned out to be an orphan receptor that is critical for controlling short-term working memory. So it's been really neat to see how some of these resources that we set up for the purposes of studying diabetes and related phenotypes have been very useful in collaborations that we would not have really been able to tackle otherwise. Another study that was directly funded by the ADA pathway support is one that again focuses on enteroendocrine cells. These are it's a rare kind of cell type in the gut that secretes hormones critical for energy balance. Many of you may know that the abundance and function of EECs compromised in the context of chronic obesogenic high-fat diet. You may also be aware that in the context of bariatric surgery, which is one of the more effective interventions for obesity and diabetes, the abundance of enteroendocrine cells are rescued. But what some of the key factors are that might be controlling EEC abundance and differentiation from naive intestinal stem cells remains definitely incompletely characterized. In this study that I'm showing you here, a graphical abstract for, we were able to identify a microRNA that is highly specific to the EEC lineage in the gut, and moreover, was sensitive to both chronic high-fat diet and bariatric surgery. And what we were able to demonstrate through functional studies ex vivo using gut organoid system is that this microRNA is controlling not only intestinal epithelial growth and proliferation, but also EEC differentiation. We have follow-up in vivo studies looking at this microRNA, and preliminary results do suggest that there is concordance with what we found ex vivo. A final example here, also led by Amy Hung. She was the lead author on the previous study and also the lead author on this one. In addition to understanding intestinal homeostasis in the adult, we've been interested in the development of intestinal stem cells. This has been notoriously difficult to study in humans, and so we've taken advantage of directed differentiation protocols established by Jason Spence and Jim Wells, and in fact closely collaborated with Jason in order to establish this in the lab so that we could define transcriptional networks that are really critical for the very early development of intestinal stem cells, as well as regional patterning. We know that intestinal stem cells that give rise to cell types like EECs are a little bit different in the duodenum, the proximal small intestine, relative to the ileum, the distal small intestine. And so we were not only able to study small intestinal lineage formation, but also regional patterning. We uncovered several different transcription factors that really hadn't been discussed previously in the context of intestinal development. Even for transcription factors like CDX2 that we had known about through decades of study in mice, we found that at different stages of development, the same transcription factor was assuming highly different cystromes or targetomes, if you will, which was fascinating for us to uncover. So this brings me to the study that I actually want to present today, which is a collaboration between my lab and the lab of Philip White. Chris just mentioned that Phil will be one of the invited speakers for the symposium on the 25th, and he joins a cadre of really excellent speakers that day, so I hope you can make it too. Phil is doing really fantastic work with his lab down at Duke, but this collaboration really would not have been possible without the programs that the Pathway had set up for awardees to be able to meet. And what I'm going to describe to you today was birthed when Phil and I kind of were walking the beaches in San Diego at an ADA Pathway event, and we came up with this idea. This is led on my lab's end by Amy Hung again. If you're a little confused why I keep highlighting Amy, She really genuinely has been the lead on all of these efforts, even though they seem quite disparate. And that's because she is a postdoc par excellence. Really, I cannot say enough good things about her as a trainee. She's on the job market right now. And if any of you know about opportunities that might be a good fit, please do let me know. I would be thrilled to pass them along to her. She came to my lab as a nutritional biochemist and reinvented herself as a functional genomicist in a matter of 3 to 4 years, with many papers to her name. So she's a really creative and prolific scientist and would be an asset in any department. I do want to highlight again that the ADA and the Pathway Program for making this collaboration possible, as well as Andrei Chernoff at Laval University in Quebec. Phil had access to a biorepository that is supervised by Andrei Chernoff, and without access to those tissue, this study wouldn't have been possible. I'm assuming most of you are familiar with both non-alcoholic fatty liver disease and non-alcoholic steatohepatitis, or NASH, but very briefly, NIDDK and other institutions define NAFLD as basically an excess of fat in the liver. Many people have NAFLD. The progression to NASH does not really occur until you have what's called hepatic ballooning, really an enlargement of the cells that is caused in large part by inflammation. There's an increase in injury and damage and severe inflammation. Sometimes NASH can progress to cirrhosis, which is defined as really extensive fibrosis and scarring that can lead to many downstream complications, not the least of which is liver cancer, and sometimes just all-out liver failure requiring a transplant. It's thought that there's a certain stage along the progression to cirrhosis where this is just not reversible. So there's been a lot of interest in trying to understand what is really driving fundamentally the progression to NASH, and how might we be able to reverse course to prevent further complication. There are currently no proven drugs to treat NASH, and so as you can imagine, there's an unmet need here that's critical to tackle. Don't hold too tightly to these numbers. Different sources will give you slightly different numbers. But the point here is that the global prevalence of NAFLD and NASH is high, and it is getting higher by the month, by the year. NAFLD was not really identified and described until around 1980, when it was realized that a massive fat accumulation in the liver is possible even without chronic alcohol use. And ever since then, the prevalence of this disease has been on the rise. There are a number of risk factors for NAFLD and NASH, as you might imagine. Obesity is one of the best discussed ones. As you can see on the right, there's a monotonic increase for both men and women in the prevalence of NAFLD as body mass index increases. There are several other risk factors: diabetes, dyslipidemia, aging, of course, and there have been, through genome-wide association studies, some genetic variants that have been identified as strong risk factors, including this particular SNP indicated here in the PNPLA3 gene. So one of the questions that's interested us, and Phil as well, is what are some of the key molecular networks that are contributing to NASH? What's different in individuals who appear to be prone to NASH versus those individuals with NAFLD who don't go on and progress to a more severe NASH-like phenotype? What are some of the key differences, and might that reveal potential therapeutic targets? And when I say molecular networks, there are, of course, a lot of different ways to tackle that. In this particular talk, I'll be talking about transcription factor networks. We also have interests in microRNA networks and signaling networks, but today I'll just be talking about transcription factor networks. We'll be leveraging a technology that was pioneered here at Cornell called CRO-seq, or chromatin run-on sequencing. It's the newest generation of nascent RNA sequencing technology. I don't have the time to get into the details, but essentially it is a technique that allows one to be able to capture transcriptionally active RNA polymerase on the chromatin. The reason why this is powerful is because transcriptionally active polymerase is found not only on gene bodies, but also, of course, at promoters and at active enhancers, not candidate regulatory elements, but those regions that are really bound by transcription factors and actively functioning as enhancers. What this means is that with one assay, we're able to identify the rate of transcription at gene loci. We're able to identify active promoters and active enhancers. And not only that, the level of polymerase transcription there is an indication of the extent of activity. So it's not just binary on or off, but we're able to tell how extensively utilized these regulatory elements are in the tissue that we're interrogating. In the interest of time, I won't go into the benefits of CRO-seq relative to ATAC-seq and RNA-seq, but I'll just highlight again that it is an assay that is able to give us three different kinds of information that usually you need to do several different genome scale studies and integrate the data to be able to get all of this information. One of the nice things about CRO-seq is, unlike the techniques from which it was derived, GROW and PRO-seq, it can be applied to archived tissue samples, which in the last couple of years has meant that biobanks all across the country and world are opening up for being able to run this technique. So here was the idea. What if we could identify from a cohort available to us obese individuals that can be grouped as either NASH resistant or NASH prone? I'll refer to these as NR and NP. We perform CRO-seq on liver biopsies from these individuals, define the chromatin activity signature, in particular enhancers, and utilize that information to be able to determine the transcription factor networks that are associated with NASH resistance and that might be associated with being prone to NASH. Again, I'll just orient you to the right panel, where in the middle section of the figure, I'm indicating again the three major pieces of information that we'll get out of CRO-seq. You'll see these peaks that are either in red or gray. These are CRO-seq read pileups that are indicative of bidirectional expression. This is the classic hallmark signature of the recruitment of transcriptionally engaged polymerase. And when you see short overlapping bidirectional expression like here, that is the hallmark signature of a transcriptional regulatory element, an enhancer in this case. If you see it near the transcription start site, it's a promoter. And then of course, across the gene body, you'll see read pileups in one particular strand. So again, these are the major pieces of information we're able to get. If we profile chromatin activity in NR versus NP, we're then set up to be able to determine what chromatin regions are the most different in terms of activity in NASH-resistant individuals. So the first step, of course, was to identify these individuals, and this is where Phil's access to the repository in Quebec was extraordinarily important and valuable. We were able to identify 6 individuals that we classified as NR and 8 that we classified as NP. So really, really important to take away from this slide is that the NR and NP individuals had almost identical levels of diabetes status, fasting glucose levels, BMI, circulating lipids, HbA1c. In fact, the NASH-prone individuals were not older, but rather younger, as can be seen in the age bar graph here. And so the only real differences between NR and NP are that there is more steatosis in the NASH-prone individuals, and then, of course, the presence of ballooning and the beginnings of fibrosis. Right? So by matching according to all of these other more classic risk factors like BMI and age, the goal was to be able to try to get independent factors that might lead to NASH resistance. Just want to highlight that these individuals were genotyped at several loci of interest, and the relative proportions of different genotypes at the most well-known risk locus in PNPLA3 were matched across these two groups, as well, again, as glycemic status. Just to validate that we really did have two different groups worth investigating further, LCMS was performed in plasma, and so we were able to look at metabolites of interest. What's being shown here are amino acids, branch chain amino acids, branch chain alpha keto acids. And what you'll see as an example is that serine and glycine are elevated in the NASH resistant individuals versus the NASH prone. So the color is indicative of a fold change in NR versus NP. The more purple, the more the level of that metabolite in the NR individuals. And you can see that, as expected, serine and glycine, for example, are elevated in NR. It has been shown in several studies that serine and glycine deficiency is associated with the development of NASH. And there are even some clinical trials in place to see whether supplementation with glycine or serine may be helpful for reversing course. But again, for us here, this was more of a validation that we had really identified two different groups. So then we embarked on leveraging the CRO-seq data to identify differentially transcribed genes between NR and NP. As you can see, we identified several hundred in this volcano plot. Every dot in this plot represents a gene. Those highlighted in purple are elevated in NR, and those highlighted in blue are elevated in NP. The pathway analysis on the right indicates that genes that are elevated in NASH-resistant individuals are enriched in pathways for fatty acid oxidation and beta oxidation, as you might suspect, whereas genes that are elevated in NASH-prone individuals are enriched in neutrophil activation and similar immune pathways, which again is exactly what we would expect given the presence of ballooning and inflammation in those individuals. So this isn't so much a new result as much as it was establishing the reliability of the data. Moving on, we then used the CRO-seq data to identify TREs, or transcriptional regulatory elements, promoters, and enhancers. And what you see on the plot on the left, each dot represents a TRE that we identified from the CRO-seq analysis. Those dots that are present on the right tail are TREs that are much more active in NR compared to NP. Those TREs that are on the left tail, on the bottom here, are ones that are much more active in NP relative to NR. We were able to identify about 1,407 enhancers that appear to be specific to the NR group, and 694 enhancers that are specific to the NP group. We did also identify promoters that are highly differentially active across groups. For the rest of this talk, though, I'll be focusing on the enhancers that we identified. So having identified these, we wanted to see whether the presence of these more or less active enhancers really were having the expected effect on gene expression in those same tissues. So what we did here is what's called an enhancer density analysis. For every gene, we identified its transcription start site, and then took a 100 kb window on either side and counted up the number of enhancers that we found that are specific to NR in this particular case. So what's being plotted immediately below the schematic here in this sort of dot scatter plot, every circle here is a gene, and the size of the dot corresponds to the number of NR-specific enhancers that are identified in the defined window. And what you'll see below is that the more NR-specific enhancers you have in the vicinity of the transcription start site, the greater the level of upregulation of that gene in NR compared to NP. This is exactly the same trend we found for the NP-specific enhancers, but just in reverse. In other words, the more NP-specific enhancers were around a gene of interest, the more downregulated that gene is in NASH-resistant individuals compared to those that are NASH-prone. What this establishes is that proximity of enhancers that are specific to one category or the other is highly correlated with the gene expression patterns that we're observing in those same individuals. So this allowed us to be able to then do the classic motif enrichment analysis to get closer to the transcriptional networks that might be underlying these phenotypes. We found what's being shown on the left and the right panel here are motifs of specific transcription factors that are the most enriched in NR specific enhancers on the left and NP specific enhancers on the right. What was most impressive to us is that most of the motifs that we found enriched in NASH prone individuals are for AP1 subunits or for transcription factors that are known to interact strongly with AP1 subunits as heterodimers. And what these stars indicate next to each of these transcription factors is that not only is their motif enriched in NP specific enhancers, their levels of expression are also significantly elevated, and not just expression, but transcription rates according to CRO-seq. What we can do with this data now is to start to look at combinations of different transcription factors. What genes are nearby enhancers that are specific to either NR or NP that have motifs for various combinations of these transcription factors? For example, SHMT1 involved in glycine and serine biosynthesis. It happens to be the only gene that has an an NR specific enhancer in the vicinity that has binding sites for HNF4 alpha and TR4. I won't say much more about this because it'll just be listing off a bunch of genes and what transcription factors they might be associated with. But we do think that there's a lot of really interesting information to mine here because we'll be able to identify different combinations of transcription factors and the downstream targets that they might be regulating. In addition to just looking at individual enhancers, we can look at what's called super enhancers. You can just think about these as clustered enhancers that have very, very high levels of activity. I won't go into in the interest of time because I'm going to wrap up in the next couple of minutes here. But we did identify super enhancers according to a methodology that is similar but not identical to the one introduced by Rick Young. We were able to establish, shown on the right panel here, that genes associated with super enhancers that are specific to the NASH-prone subset are indeed downregulated relative to NR much more significantly than those genes that are not associated with NP-specific super enhancers. Again, this is exactly what we would expect if indeed these super enhancers are exerting particularly high levels of control on neighboring genes. If you take a look at these individual genes that are nearby to NP-specific super enhancers, again, we were struck by how many of them are related to AP1. You'll see FOS and MAFK, JUNB, FOSB. And so this led to the beginnings of a working model where we think that NP-specific super enhancers are driving the upregulation of a lot of AP1 subunits that are in turn binding to NP-specific enhancers and driving pathways related to inflammation, chemokine regulation, and ROS regulation. So this is the last slide. I just want to mention that CRO-seq is not sensitive enough to be leveraged at a single cell level. So what we have now is bulk tissue signal, and AP1 has been associated with fibrosis. So there's a possibility of the involvement of hepatic stellate cells. It's involved in inflammation, so all kinds of immune cells and recruited neutrophils might be relevant here. Kupffer cells themselves might be relevant. We really want to start to build a single cell enhancer profile. And so we are now working toward doing single cell attack-seq on these samples to deconvolute the CRO-seq signals and build cell type specific enhancer profiles so that we can determine in which cell type is which of the AP1 subunits most active and what are their targets. With that, I will thank my lab and all of their support in this and related works, and again, particularly highlight Amy Hung for all of her leadership. thank Phil White for his really wonderful collaboration here, and the ADA Pathway Program.
Thanks very much, Praveen. That was exciting and really clear. Like me, you took essentially all of your time, which I always do as well. Just two, I have a question, and there's two on the chat very quickly. So given that one of the selection criteria for your NASH prone versus NASH resistant is ballooning and inflammation markers, how do you sort out cause and effect when you get these networks? That's a tough ball to unwind, it would seem.
It is, you know, and I should make it clear, I'm not at all suggesting that the discovery that inflammatory pathways are coming up is new in and of itself. I think the new layer of information here rests in part on the specifics of where these factors are binding and eventually in which cell types they might be binding, so that we can start to tease apart the networks. As far as cause and effect, I think one of the things that we really need to move into is to be able to get progression. And I think this is going to be difficult with samples. But to the extent that is possible from these cohorts that we've been lucky enough to tap into, we'd like to try to identify individuals along the phenotypic progression and then see if we might be able to identify pathways that are kind of on the way.
Okay, let's see if you can answer a couple questions rapid fire, okay? Paul Cohen, great talk, Praveen. Given the natural heterogeneity one typically sees with human studies, how consistent were your findings within subjects in each group? Quick answer.
Actually, remarkably consistent.
Okay.
But of course, there's 6 and 8, and we need to grow the cohorts.
Okay, Steve Rich, do you see different cross-seq signals during development in different tissues? And are there any issues of differential splicing in CRO-seq?
Yeah, so different CRO-seqs during development. Do you mean development of the disease, Steve, or during actual development?
Actual development, but also particularly once they get disease, can you see something pre and post disease development?
Absolutely. Yeah, we see really, really strong differences in both ways, during development as well as upon development of disease.
Okay, Andy Stewart, how well do CRO-seq identified enhancers and promoters correlate with the same elements identified by methylation marks, ATAC-seq, and do patterns of CRO-seq differ between pre and post bariatric surgery biopsies? That's an interesting question.
Yeah, so the latter question is something we're tackling right now. I'm sure it will, but I don't have a precise answer, but it's very, very exciting, and I love the question. The CRO-seq based TREs that we identify are highly overlapping with ATAC-seq, but the advantage is that in ATAC, you're getting all of the candidate regulatory elements, and this is essentially a subset of those that are actually active at that snapshot in time.
Okay, there's also a question from Steve Parker and Stitzel, your colleague, but we're gonna have to move on in fairness to Sarah. So Sarah, if you could put your slides up, please. And Praveen, you'll be able to see those questions in the chat. So please answer the questions we didn't get to, okay?
Absolutely.
All right, and then we have Sarah Tishkoff from Penn. She got a Visionary Award in 2019, and she'll speak about impact of subsistence and genetics on cardiometabolic traits in ethnically diverse Africans. Sarah, please.
Great, thank you. Praveen, you're a really tough act to follow, I have to say. But now for something completely different, I'm going to start by talking about what it is that motivates my research. And that is to address several key challenges in human genetics research. The first is we need to do a much better job characterizing genomic and phenotypic variation in ethnically diverse populations across the globe. We want to understand the evolutionary processes that generate and maintain that variation, and to understand how gene-protein, and gene-environment interactions contribute both to normal variation and disease risk. The focus of my research is on Africa, and there are a number of reasons for that. One of which is the fact that all modern humans originated in Africa. These red dots represent the locations of fossils of anatomically modern humans. the oldest of which was dated to about 300,000 years ago. Following this origin in Africa, sometime in the past, say 50 to 80,000 years, there was migration of relatively few numbers of people across the rest of the globe. And we now know that when modern humans left Africa, they ran into archaic populations like Neanderthals and Denisovans, and they interbred to some small extent, such that around 2 to 6% of non-African genomes derive from these archaic populations. So the point is that this evolutionary history has shaped the pattern of variation that we see in populations today and has resulted in the highest levels of genetic diversity being in Africa. The other thing that's motivating our research is to understand the causes of health disparities. So for example, hypertension is more common in people of African ancestry relative to other ethnic groups. Type 2 diabetes is more common in people of Native American and African ancestry compared to other ethnic groups. Undoubtedly, there are socio-demographic dietary factors that are playing a major role in these health disparities, but it's thought that there are underlying genetic risk factors as well. And in fact, in a study of people who self-identify as African American, the percent African ancestry positively correlates with the risk for type 2 diabetes, suggesting there is a genetic basis to this. And furthermore, you will often see both hypertension and diabetes co-occurring. And yet, despite the importance of Africa, there's such a major bias towards including Europeans in genome-wide association studies. In this paper, we show that the GWAS at that time included about 80% of the individuals included were of European ancestry, about 10% Asian ancestry, 2% African ancestry, 1% Hispanic ancestry, and less than 1% everyone else. And that's a huge problem. It's going to impede our ability to fully understand genetic risk factors for human disease, and it could exacerbate health inequalities. Our ability to translate genetic research into clinical practice or public health policy could be incomplete, or even worse, it could be mistaken. It could result in inaccurate assessment of pathogenic variants in clinical genomic studies. And one of the reasons for this is that one of the criteria for determining if a variant is pathogenic is how common it is, with the idea being if it's really common, it's less likely to be pathogenic. Maybe it's not common in Europeans, but it's common in other ethnic groups. And then lastly, attempts to use estimates of genetic risk. So for example, polygenic risk scores to determine risk for diseases, say type 2 diabetes, for example. Almost all of these have been conducted in European-based studies. And what we know is they don't transfer to other ethnic groups. So if your patient is from a different ethnic group, not European, then the prediction accuracy is extremely low. So to overcome these biases, together with a number of African collaborators throughout many different countries in Africa, we have been doing field work in Africa, mainly focused on studying minority populations who practice traditional lifestyles. And I thought I'd show you a little bit of what that's like. These populations live in more remote areas, so it requires bringing a four-wheel drive vehicle to get there. We're really careful to do this research in an ethical manner. So that means not just getting IRB approval, but going through very rigorous ethical review in each country. After we get a permit, we also spend a lot of time doing community discussions, translating into the local language and using layman's terms to explain the projects. And only after we get community and individual consent do we proceed. Also really important is returning results to participants. To me, it's a form of benefit sharing. And we translate we summarize in layman's terms and translate into the local language. Training and capacity building is really important. And I've had the honor of being able to train a number of grad students and postdocs from Africa. Mamadou is the latest, will be coming this summer. And the hope is that they are going to go back to Africa and that they will enable research on the continent. So in these regions, we're getting blood we started using liquid nitrogen 2010. So after doing that, we can now preserve RNA, we can freeze plasma. We get very detailed ethnographic information, any information about diet that they might have, any medical information they may have, though it should be noted that they're they have very limited healthcare in these regions. And then we have to process these samples in places where there's limited or no electricity. So in the lower left, we've started bringing a generator and we just set up the lab in the bush. So then we measure phenotypic diversity in a non-invasive manner. We're not doing a disease-focused studies in the past. This has been just looking at the general population. We look at very detailed anthropometric phenotypes, not just height and weight, but waist circumference, percent body fat, and so on. We look at cardiovascular, lung, and blood phenotypes. We look at metabolic function, lactose and glucose tolerance, and infectious disease status when we can. And then we're using an integrative omics approach, collecting different types of omic data, and then integrating that, looking how these are impacting each other, and also how environment and things like diet are impacting this data and ultimately together impacting these variable traits and disease risk. So I want to start by telling you a bit about the patterns of variation in Africa. And this was from a study we did a while ago, but it's the largest study of genomic diversity across ethnically diverse Africans, even to date. So we studied over 3,000 Africans and we inferred their genetic ancestry, which is shown by the different colors. Think of this like a kind of like a 23andMe test that you might get. And we just pooled individuals by region. But the point is, you could see by all these colors that there is a huge amount of genetic variation, not just within, but between populations in Africa. And groups in Eastern, Southern, Western, Central, and Northern Africa are very divergent from each other. And there's a lot of admixture within each region. And if you look at the faces here, you could see there's a lot of phenotypic variation. And this is reflecting the evolutionary history of Africa and adaptation to very diverse environments and diets. So more recently, we've generated high coverage whole genome sequencing from 180 individuals shown here. And they were selected to represent sort of all those different colors, the most ethnic diversity. I also just want to mention when I show you our lipidomic study, these are largely the populations that I'm going to be presenting there. So from sequencing 180 individuals, we found millions of single nucleotide variants, and about 17% are novel. They're not in any current database. Many of those are predicted to be functional, so they're either altering amino acids or they're in predicted regulatory sites. So we can imagine if that's just 180 individuals, we're going to find a lot more when we increase the numbers. From this data, we can make a phylogenetic tree. At the end of each of these branches is an individual. We've labeled them by the ethnic group. And what we see is that the oldest lineages, those that are basal at the root of this tree, are the two San, Khoisan speaking groups, hunter-gatherers from Southern Africa, followed by the Baka, who are so-called pygmy hunter-gatherers from Central Africa. And then we see clustering by region, West Africa, East Africa, and then non-Africans who basically have a subset of the African diversity. So now I want to tell you what the focus has been of my Visionary Award, sort of what motivated me to begin with. And it had to do with when we were doing fieldwork in Cameroon. And we were studying a group called the Fulani, who are traditionally practice a pastoralist lifestyle. And we could see from these colors here that their ancestry is quite different from neighboring Bantu speaking agriculturalists shown in orange or pygmy hunter-gatherers shown in green. And what stood out to us was the exceptionally high incidence of hypertension in adult onset diabetes. Now, 8%, 5 to 8% might not seem mind-blowing to you guys, but in traditional indigenous populations, we rarely see diabetes. And so that really stood out. And what was unusual is that they didn't have any obvious risk factors. Their average BMI is only 19. So we tested for autoantibodies, and we did rule out the possibility. It's not a type 1 diabetes. It's probably not a type 1.5 diabetes. What is it? We still don't know. There may still be some sort of an immune component. And what's also really unusual about the Fulani is that they're relatively resistant to malaria relative to other ethnic groups in Africa. And nobody knows yet the reason for this. But it's been proposed that they may have a boosted inflammatory immune response. So my hypothesis is that if they have a boosted inflammatory immune response, that's protective against infection. So these variants may rise to high frequency, but they may put them at risk for hypertension and for insulin resistance. Now, this work is of relevance to African Americans who trace their ancestry predominantly to Western Africa. And in a prior study, we showed that African Americans have as high as much as 14% ancestry from Fulani. So whatever we find the Fulani could have a lot of relevance to individuals in this country. And in addition, because these groups live in these rural environments, the environments within a population are very, very homogeneous. They're not going out shopping. They eat the same foods and so on. So we could compare people of the same genetic ancestry in Africa, say to those in the US, and try to distinguish genetic from environmental effects. So we only have a relatively few number of these Fulani. We had 400. Our goal was to go back and do an expansion of the study, continue to study. Our bags were packed, we were ready to go. This was last March, and literally we got shut down due to COVID. I don't predict we can get back there for another year, so because it has to be after they're able to get most of the people vaccinated. So we're changing track a little bit. And what we're going to do is look much more broadly across Africa and look at genetic factors influencing normal cardiometabolic traits, which is important because if you can understand that, you could also understand why some people are prone towards disease. So I want to start by telling you about a study of some of these traits that we collected, anthropometric and cardiometabolic, to give you an idea of the variation we see. And this is work that was done by Matt Hansen in my lab. This is the distribution of the populations in Africa. And he integrated these with data from an Illumina arrays. About 2,000 individuals had genetic data. And these are from groups with really different diets. So hunter-gatherers have a diet that's heavy in fruits and nuts and tubers, and to a lesser extent meat. Pastoralists have a diet heavy in milk and blood, and to a lesser extent meat. And agriculturalists have diets that are heavy in grain and corn, vegetables, fruit, and tubers. And so what I'm showing you here are this, we're only looking now at women, and we've color coded the population based on the subsistence, correlates with their diet. If we look at height, we can see that at the low end are the hunter-gatherer populations, and at the very low end are the Baka pygmy population. At the high end of height are some agriculturalist groups and the Herero pastoralists. At the low end of the BMI is this pastoralist group from East Africa. They tend to be very tall and thin body type. And at the high end are the food producing agriculturalists and the Herero. If we look at waist circumference, it's quite striking. All of these groups at the low end are all San, Khoisan speaking groups, hunter-gatherers from Southern Africa. And at the high end are two groups from Botswana, agro-pastoralist group Botswana and Herero pastoralists. And if you look at percent body fat, again, some of the hunter-gatherers on the low end, the agriculturalists and the Herero at the high end. I'm showing you a picture of a Herero woman. And one thing that's unique about them, as part of their culture, they desire women to be very overweight. So it's considered a sign of fecundity. And that's why it wasn't surprising to me that they ended up at the very high end of the percent body fat. But if we look at things like C-peptide, for example, that again is lowest in many of the hunter-gatherers, highest in the food-producing agriculturalists. Glucose, we don't see a huge amount of variation, but there's certainly a trend towards being lower in the hunter-gatherers and higher in the agriculturalist populations. So what Matt did is he inferred genetic ancestry as shown by these different colors for each individual. And he wanted to address the question, how much of the phenotypic variation that we see is due to genetics, shown in this dark gray, and how much is due to environment, that's sort of the open bar here. And within the genetic component, how much is due to ancestry, and how much is due to within population variation? So what we see, maybe not surprisingly, is that skin color is a trait that has a very strong genetic component. And furthermore, it's strongly correlated with ancestry in African populations. We see something, also a strong genetic component for anthropometric measurements like height and knee height and arm length. I was surprised to see this also for weight, BMI, and percent body fat. We see it also for LDL, HDL, and cholesterol and triglycerides, not so much for some of the other things, including glucose and C-peptide levels. So we then wanted to delve in a bit more about genetic and dietary factors influencing lipids. Why is this important for diabetes? This was from a recent review, and as they pointed out, atherosclerotic cardiovascular disease is the principal cause of death and disability among patients with diabetes. And almost 70% of patients with type 2 diabetes suffer from dyslipidemia, which represents the leading cause of cardiovascular morbidity and mortality in diabetes. So to pursue this question of what's the impact of diet and genetics on lipid patterns in Africans, we collaborated with Chuck Brandt at the University of Michigan and a number of colleagues at the University of Pennsylvania. So we looked at two different cohorts, and the first one consists of 425 individuals from 9 ethnic groups in Eastern and Southern Africa. This is a principal component analysis based on genotype data, just to show you how divergent these groups are. They are really, really genetically divergent from each other. So Chuck's group was able to identify, using a targeted lipidomics approach using mass spectrometry, 651 lipids that could be generated or put into 19 lipid classes that are shown here. Not surprisingly, when you look at the levels of these lipids, many of them are correlated. It's not a surprise because many of them are part of the same metabolic pathway. Now, we also looked more recently at 215 individuals from Cameroon. These include Pygmies, the Baka and Bajeli, some Bantu agriculturalist groups, and then these are the Fulani over here. The technology is improved, so we're able to now detect over 1,000 lipid species, and these we can place into 23 classes of lipids as shown here. So if we do a principal component analysis based on the overall lipid levels, and this is just looking at the Eastern and Southern Africans, and we've color-coded people based on their subsistence, which is correlated with their diet. One thing we can distinguish are the pastoralists. We can pretty well distinguish them, and also to a lesser extent, people who are agriculturalists. For the others, there's a lot more overlap. And when we repeat this for the Cameroonian samples, one thing that really stands out here actually are the Fulani that are shown in blue, that are very distinct. But we were able to identify, we could identify specific lipids that are significantly different and correlated with particular subsistence patterns. So this particular free fatty acid is higher in agriculturalists compared to other groups, and this particular triglyceride higher in pastoralists than other groups, and so on. And then we were able to do what I think of as a natural experiment. And what we could do is we could compare, for example, these two groups from Ethiopia. These are all Ethiopian populations. The Kiamant are agropastoralists and the Weto are foragers. actually have a diet heavy in fish. And you could see that their genetic ancestry is very similar. So genetic ancestry is similar, diet is very different. We can distinguish a number of lipids that are significantly different between these two. And in the principal component analysis of all the lipid data, they can clearly be distinguished. So that would make you think diet is really important. That's having a major role. And if we compare the Mersey, who are pastoralists, and the Shabu, who are foragers, they have also similar genetic ancestry and can clearly be distinguished here. So again, this is looking like diet is playing a major role. But then we were surprised to see the following, which is when we compare the Mersey pastoralist to the Hamer, and here you can see the Hamer have a very different genetic ancestry, but they have a similar diet. They're both pastoralists. Again, we can distinguish between these two. That would suggest that genetics is having an important role. So we want to try to distinguish what is the impact of genetics. And to do that, we integrated this data with over 7 million single nucleotide variants across the genome and conducted a genome-wide association study. We identified 324 lipid-associated genetic variants, and as shown here, the majority of these are in non-coding regions and they're in predicted regulatory regions. So relatively few are non-synonymous variants. And what was really striking is that many of these have not been previously reported. Now, the nearby regions have been reported for many of these, but the particular variants have not. We think that we're better able to find map in these populations. And as Chuck Barant has pointed out, when he did the lipidomics analysis, he said it was the most clean data he's ever seen because unlike our US population, they're not eating all this fat and certain types of fat that's swamping out the lipidomics analysis. So you get these like very fine scale level, low levels of lipids. And we were able to replicate the strongest associations in the Cameroonian data set. Now, what was interesting, I just want to give you a couple of examples. Here's an example of GWAS results for this particular lipid. And we find a very strong signal at the FADS2 locus. We could see that it looks like there are other regions of the genome that are also contributing to levels of this particular lipid. So it's not just monogenic. Now, if you look at these two groups of the very different diets, the Kiamont and the Wato, we're plotting here the relative amount of this particular amino acid, controlling for BMI and age. And while they're very different, which we think is probably due to the very different diet, within any population, they are clearly impacted by the genotype at this FADS2 locus. And if we look more broadly, again, you could see these differences between groups may be influenced by diet, but within a population, they're all impacted in the same direction by the genotypes at this locus. This is also clear at another locus. We don't know the target gene, but associated with this levels of this particular lipid. And here we see that this lipid is very high in the pastoralist groups and very low in the hunter-gatherer groups. Again, but within the populations, very impacted by genotype. So the take home, I guess, is that genotype, genetics, and diet are playing a major role. And we're still trying to figure out, disentangle the proportion of the variants due to diet and that due to genetics. So in the future, we're going to be analyzing untargeted metabolomics data sets in the same individuals, generating data on cardiometabolic biomarkers in a much larger set of samples. As part of the TopMed consortium, we've generated about 6,500 high coverage whole genome sequences that we're integrating with this data to do GWAS. And then we can integrate it with the larger TopMed data set, which has a large number of people of European ancestry and African American ancestry to determine if these associations replicate and to try to develop more effective polygenic risk scores. And then lastly, we're characterizing differences in transcriptional and epigenetic response to immune challenges in white blood cells using single cell sequencing techniques to identify genomic and epigenetic variation contributing to differences in immune response between populations with differing prevalence of diabetes, such as the Fulani. We think that might give us a clue about what is happening in that group that makes them predisposed to diabetes. So with that, I will just end by thanking everybody who's contributed, and of course the funding organizations, particularly my Pathway Award. Thank you. I'm happy to answer any questions you have.
Thanks, Sarah. That was terrific. I don't see anything on the chat quite yet. Please go ahead. But I'm gonna grab at something that was sort of midway in your talk before you got into this beautiful integration of genetic and metabolomics analysis. But you said that it's possible that a gene has evolved to provide protection against disease caused by mosquitoes. Mm-hmm. And I was struck by the similarity of Opi Olabisi's work. He works here in our institute, where there have been genetic mutations that protect against trypanosomes, yes, which end up causing disastrous kidney disease in African American subjects. So that's an instance of actually a single gene mutation. Mm-hmm. Because it's the ApoL1 gene and it right. appears multiple times. Mm-hmm. Do you anticipate something like that when you are able to get back to it, coming out of that phase of your work as well? Yeah, that is exactly the hypothesis, that just like sickle cell, and in this case, ApoL1 right. is strongly associated with risk for kidney disease in people of African ancestry. And right. they found that these mutations in ApoL1 show a very strong signature of natural selection. So there's something adaptive, there's something important about them, and they rise to high frequency. And it turns out they're protective against trypanosome infection. Exactly. And that's exactly what I think. I mean, it is a huge mystery. Nobody can figure out why the Fulani are resistant to malaria. It's not because they have sickle cell. It's not because they have the typical, known mutations. Something else is going on in that population. And then the other thing, I was just blown away by the high incidence of hypertension and adult onset diabetes. And when we went there, they brought this up, and they are not quite as remote as some of the other groups. So they do get some clinical attention, and many of them were on metformin and were asking us, please help, try to figure out what's going on here. Now, they are a founder population because the Fulani are traditionally nomadic. They would walk their cattle across Central and Western Africa. And these guys settled in 1930s or 40s in that area in Nigeria. So there could be a very strong founder effect. Yeah. So that could have contributed to the high incidence of this. Yeah. But that's why when we go back, what we wanted to do is to study the Fulani that are in Eastern Cameroon, who have a more traditional lifestyle, to see if they also are having this high incidence of hypertension and diabetes, but can't do it until maybe next year. Okay, so just time for just one question from Rich Bergman. Do you think beta cell dysfunction or insulin resistance will be the pathogenic path in the Fulani? Are there any defects in insulin clearance, as are found in American Blacks, that you've observed so far? Oh, and this is where I'm going to have to express my ignorance, because I'm a human geneticist and I'm not familiar with all the testing. Right. I would like to know, how would you distinguish that, if somebody could tell me? Rich, are you on? Do you want to speak to that? Can you turn yourself off mute?
Well, I'm on now.
Yes.
I think since you've measured C-peptide, which is great.
Not yet. We haven't done it yet in that group. We're just about to do that.
But if you do,
yeah,
then I think that we probably could do some surrogate markers and try and answer that question, because it's very rare that people with that BMI have diabetes, as you know.
Yes, exactly.
And so it really looks like a beta cell defect, and I would not imagine that they're insulin resistant. So I think doing phenotyping would be very, very interesting. And we'd be happy to work on that if you're interested.
Oh, that'd be awesome. Yeah, I definitely will follow up with you.
Great.
I was
going
to
very interesting talk.
With that, two scintillating talks today. Thank you to Praveen and Sarah. Reminder: June 25th, 10 to noon will be the ADA Pathways Symposium, and then our next in this series is July 7th Stephanie Stanford and Matthew Weber will speak on July 7th at 2 o'clock, our usual Wednesday session. Thanks, everybody. Thanks to the speakers. Great session. Cheers, everyone.
Ciao.